All right, so before I get started talking about the endocrine system, I want to do a review of receptors and hormones. We talked about this a little bit in ANP1, but I know that it's been a long time since then, and really, when you're talking about the endocrine system, what you're talking about is hormones and receptors and how those hormones control cells. And so it's really vital that you have an understanding of what receptors are, how they work, what hormones are, how they work, and what factors can affect the hormone receptor interactions. So first off, let's say, what is a receptor? All right. Receptors are almost all proteins, and they bind very specifically to a thing. That thing that they bind to is called a ligand. A ligand is a general term for anything that binds specifically to anything else, but usually a protein. When a receptor binds to a ligand, it usually produces a response. There's no real point to bind something if you aren't going to react to it in some way. That response could be a bunch of different things. We'll talk about what some of them are later. But it produces some change in how the cell works. That could be a long-term change. It could be a short-term change. It could be a morphological change, but it produces some sort of change in how the cells work. Receptors are a cell's eyes and ears on the outside world. Right? Everything that a cell knows about its external environment, it's probably getting that information from some sort of receptor. Some of that information is going to be like practical stuff like, the pH, or some sort of, maybe if we're talking about an amoeba, maybe it's receiving some sort of chemicals to track its prey. Most receptors, not all of them, but most receptors are found in the plasma membrane. They are intended to tell the cell about the external environment. There are some receptors that are found inside the cytoplasm. Those can only detect things that can cross through the plasma membrane. Or there are some receptors that actually tell the cell information about what's going on inside of the cell itself. Those type of receptors are not usually involved in the endocrine system. But the ones that can detect hormones that can cross through the plasma membrane are part of the endocrine system. To remind you what the plasma membrane is, the plasma membrane, which is found in all cells, is primarily composed of phospholipids. Phospholipids have two portions to them. They have a hydrophobic portion, which are fatty acid tails, and they have a hydrophilic portion, which is a charged phosphate group, and they are arranged in a bilayer so that the fatty acid tails face each other, and the phosphate head groups face the water outside the cell or the cytoplasm inside the cell. And the plasma membrane, by its nature, is selectively permeable. That means that some things can cross it, and other things can't cross it. Basically, anything that is hydrophobic can cross through. So we've got some hydrophobic molecule out here, and it can just cross through into the cell. Let's say you have a hydrophilic molecule. Hydrophilic molecules don't like being in hydrophobic zones, so they tend to not cross this barrier very easily. Unless there's a protein, like a channel protein or some sort of transport protein, that will specifically admit them. So let's say we have a transport protein here, like maybe it's an ion channel, and now it opens up, and the hydrophilic thing can now cross through. But hydrophobic things usually don't need any assistance passing through the plasma membrane. Now, sometimes they need assistance getting to the cell because the blood is primarily water-based, and hydrophobic things don't like hanging out in water. So many hydrophobic chemicals, lipids, steroid hormones, cholesterol, things like that, they often don't do well by themselves in the blood and have to have special carrier proteins, things that bind and protect them from the watery environment, and then drop them off wherever it is that they're going. So like we said, most receptors are found in the plasma membrane. The vast majority of them are. And they typically face the outside environment where they can bind to some sort of signaling molecule, be it a hormone or something else, and then relay that signal inside the cell. Those are membrane-associated receptors. They will typically respond to hydrophilic ligands. Intracellular receptors, all right, these are going to be receptors that are found inside the cell, and that means that whatever the signal is, whatever they're detecting, has to be able to cross through the plasma membrane, meaning that it's usually going to be hydrophobic. Often we have a carrier protein which carries this hydrophobic molecule to the vicinity of the cell, and then once it's dropped off, it just diffuses into the cell where it binds the intracellular receptor that then responds by doing whatever it is that that signaling molecule tells it to do. Most hydrophobic signaling molecules are a class of hormones called steroids. And steroids often have very long-lasting effects, modifying what genes the cell is expressing. So they're slower, but they last longer than most other hormones. So how do receptors work? There's actually a bunch of different ways that receptors work, and you don't need to know all of the various mechanisms. But they all work off of a few similar, at least all of the membrane receptors usually work off of a few similar concepts. First off, it's a three-stage process. There's reception, where the ligand binds to the receptor. There's transduction. The receptor itself usually does not produce the final signal. It's like a relay race. So the receptor hands off the signal to another molecule, that hands it off to another molecule, that hands it off to another molecule, and eventually one of these molecules is the molecule that is responsible for doing stuff. And whatever the change that happens in the cell is called the response. So here you see a receptor system. The ligand here is usually called the first messenger, and you'll see why in just a second. The first messenger, or ligand, binds to the membrane receptor. The membrane receptor usually responds by undergoing some sort of change. Maybe it changes its shape when it binds the ligand, Maybe it phosphorylates itself. Maybe it activates some sort of catalytic function that it has. In one of the most common types of receptors, what it does is it's normally bound to this protein called a G protein. But when the receptor binds to, or when the ligand binds to the receptor changes shape so that it releases this G protein. And then the G protein is now free to move around and diffuse throughout the membrane, and it binds to an effector enzyme called adenylate cyclase, typically. And this effector enzyme makes what's called the second messenger, right? Because, remember, we're talking about a hydrophilic message, so the first messenger can't get inside the cell. The second messenger is what's going to carry that signal all throughout the inside of the cell. And in this specific example, the second messenger is what's called cyclic AMP, which is something that is, it almost never occurs by accident, and it's very unique looking, but it's also very easy to make from a very readily available molecule, ATP. All cells have ATP, so it's pretty easy to find some ATP that you can then convert into this very specific second messenger molecule. This cyclic AMP, the second messenger, undergoes, it binds to a protein and activates that protein. That protein then goes and modifies a bunch of other proteins, which modify a bunch of other proteins, which then go and actually change the cellular environment. This is what's called a signal transduction cascade. And its purpose, or one of its purposes, is amplification. All right, so you can see here there's one ligand binds to one receptor, right? So normally that would produce a very, very small response. But because of this amplification process, right, so the G protein relays its signal to this enzyme doesn't just make one second messenger. It's an enzyme. It's a catalyst. So it can make hundreds or thousands of second messengers. So now this one first messenger has been turned into thousands of second messengers. Each of those second messengers is going to bind to a protein. Those proteins are also enzymes, and so they're going to alter potentially tens of thousands of third messengers. And then those activated third messengers are going to act on another protein, and basically, because at each step, right, whatever the actor is going to do its job more than once. It's going to make more than one second messenger. It's going to turn on more than one protein. That means that at every stage of this relay race, where the signal is being transferred from second messenger to third messenger, on and on down, the signal gets amplified by many times. That's how one or relatively few ligands binding to a cell can produce such a dramatic and fast response inside of the cell because of this signal amplification. If we're looking at a steroid hormone, so a hydrophobic hormone that's going to pass through the cell membrane, usually it binds to a steroid receptor that's inside of the cytoplasm. And together, this what's called hormone receptor complex, once the hormone receptor complex is complete, it gets moved into the nucleus, where it will typically turn a gene on or off. Note that you don't get that signal amplification. You don't get that second messenger enzyme cascade. This is one of the reasons why steroids are usually slower response you do eventually get some very dramatic changes because the cascade, the amplification actually happens after the hormone reaches the nucleus, because when this hormone turns a gene on, you're not just going to make one copy of that gene. You're not just going to make one protein of that gene. You're going to make hundreds or thousands of them, and you're going to do it for a long time, for as long as that gene is activated, which is usually for as long as the steroid is present. But they are a little bit slower getting off the ball. So what are the effects? We've talked about reception. We've talked about the transduction. What are the effects that ligands can produce? Well, there are a ton of different effects that ligands can produce. In fact, pretty much think of anything that a cell can do. That's something that a ligand can trigger the cell to do. But there are some more common things that they frequently do. You can turn enzymes on or off. So an example of this would be the insulin, right? So insulin tells cells, specifically cells in the liver, when insulin binds, it tells those cells to turn sugar that it's now importing. It's now grabbing that sugar from the blood, and it tells it to turn that sugar into glycogen. So it activates an enzyme called glycogen synthase. All right? Ligands can open or close channels in the membrane. If you think about how muscles and nerves work, the ligand-gated ion channels are a good example of this. Ligand binds, membrane channel opens, it allows in the ion, or whatever it is that it's supposed to allow in. Receptors can change what genes are being expressed. We saw an example of that with our steroid hormone on the last slide. Even non-steroid hormones can sometimes have changes in what genes are being expressed. For instance, thyroid hormone and growth hormone are both good examples of that we'll talk about later. Receptors can cause morphological changes in a cell. Like a good example of this is in the cells of the immune system. When you are damaged or infected, you produce a chemical called an alarmone, which basically just means a warning hormone. And one of the things that does is that it causes the cells of your blood capillaries to change shape. It changes them from being fairly tight, flat cells, squamous cells, to being more permeable, looser bound columnar cells. And what that does is it allows more things to leak out of your blood vessels. It makes them become more leaky, and that's important because you have white blood cells that are going to be coming to that area to fight off the infection. And those white blood cells normally don't leak out of the blood vessels very easily, but you need to cause this morphological change, which will cause them to leak out in the right place where the infection is. Receptor effects can also cause endocytosis or exocytosis. Exocytosis, you're all very familiar with how nerves work. The binding of the receptor triggers a cascade of events. You know, the ions come in, you get action potential that moves down the nerve axon, and then when it reaches the end of the nerve axon, it triggers the release of your neurotransmitter through exocytosis. Your neurotransmitter is sitting there in these little vesicles, and it just basically it allows them to. So let's go. Here we've got our little nerve axon, and in the bulb there at the bottom, you've got your vesicles, and when the signal reaches the end, these vesicles sort of fuse with the membrane and release their neurotransmitters into the environment. You can also cause receptor-mediated endocytosis. There are a lot of things, proteins like that are often taken into the cell because they bind to receptors, and then once enough of them have bound to those receptors, just the whole area that those receptors are in just sort of bubbles off inside the cell and takes in whatever was bound to those receptors. This is actually a common way that viruses can actually exploit your body. They pretend to be something else, they bind to these endocytosis receptors, and then your body thinking that it's taking in something good, something healthful, will actually be importing this virus into the system. So far, we've talked a bit about hormones, but mostly we've been talking about ligands. So, receptors bind ligands, but what is a hormone specifically? Well, all hormones are ligands because they all bind to receptors. A ligand is anything that binds to a receptor. But hormones are a specific class of ligand. Hormones are ligands that are specifically produced by the body to convey a specific signal. And hormones are usually secreted into the blood and convey information through the blood over long distances to produce responses. So, basically, while lots of things can serve as ligands, for instance, glucose is a ligand to cells in your pancreas, right? They have to be able to detect what your blood sugar level is. So they have receptors for glucose. And when they sense that there's a lot of glucose in your blood, that triggers your pancreatic islet cells to release insulin, right? So that's an example of a ligand producing a response. Your pancreatic cells monitor the external environment. Glucose, however, is not considered a hormone because it's not something that is produced as a signaling molecule by the cell. It's just something that it's detecting in its external environment. So hormones are always things that your body intentionally manufactures. There are 4 types of signaling molecules, or at least 4 classes of signaling molecules based on how they are excreted and what distance their effects have. These are all going to be crins, right? So exocrine is the first one up here. Exocrine are hormones, or at least they're things, chemicals, that are secreted through ducts to the outside of the body. So if you think about your sweat glands or your sebaceous glands on your skin, those are glands. Those are called exocrine glands because they're secreting substances onto the surface outside of your body. Those are usually not considered to be hormones. You know, your sweat and your skin oil is not considered hormones because, sort of by definition, if you're secreting it outside of the body, then it's not going into the blood and it's not having an effect on things inside of your body. However, there are a few specific instances where you might want to consider them sort of hormone-ish. You have to also remember that technically the inside of your lungs and the inside of your intestines and your stomach is outside of your body. There's a tube that runs all the way from your mouth all the way to your anus. Things can pass through that tube. Food comes in one side, poop comes out the other. It's never technically entered your body. It's moved through the hollow tube that goes through you. So, your gallbladder that secretes bile salts into the intestines, your pancreas that secretes digestive enzymes into your intestines. Those are all exocrine glands. And while they are exocrine glands, they are affecting your body processes to a certain extent, right? Not technically inside of cells of your body, but certainly they're affecting something that your body is doing. So, under certain circumstances, they might be considered not true hormones, but sort of hormone-ish. An important distinction to keep in mind is that all exocrine glands secrete through a duct to the outside of the body. So they're all ducted glands. Paracrine signaling molecules. This is when a cell is going to release some sort of chemical into its environment. That chemical will diffuse away from the cell and bind to other nearby cells. So here you see this cell here. It's secreting these chemicals, and those are binding to nearby cells. So because they're only binding to nearby cells, they have very localized effects. So one cell can affect just basically the cells that are close to it. Chemicals, ligands that are secreted by paracrine signaling are often not considered to be hormones, but then again, they sometimes are considered to be hormones. It really depends on who you're talking to. Like, pure endocrinologists really want to say, oh, if it doesn't go into the blood, then it's not a hormone. But they're very hormone-like things, and often they're the same chemicals that are hormones under other circumstances. But you could also consider neurotransmitters to be paracrine signaling, right? You're releasing a signaling molecule that's going to affect only nearby cells. And usually, neurotransmitters are not considered to be hormones, even though many neurotransmitters are actually the same molecules as are used as hormones in other parts of the body. For instance, epinephrine is a neurotransmitter, and adrenaline is a hormone. But epinephrine and adrenaline are actually both the same molecule. Paracrine signaling is a type of signaling that you should know. Are paracrine chemicals hormones? Not technically, but they're very, very hormone-ish. A modification of paracrine signaling is actually what's called autocrine signaling. This is where a cell secretes chemicals into the outside environment that then bind its own receptors and change the cell that secreted them. Autocrine signaling is often combined with paracrine signaling, right? A cell will want to modify the cells in its immediate environment. Also, it may want to modify itself. So often you will find a cell engaging in both paracrine and autocrine signaling at the same time. Last, we have endocrine signaling. With endocrine signaling, you are secreting your signaling molecule directly or indirectly into a blood vessel. And of course, blood goes all over your body. Pretty much every part of your body has contact with your blood because it needs oxygen. And that means that once you have secreted a hormone, and these are proper hormones, into the blood, it has system-wide effects. It can get to any cell that has the appropriate receptor for it. Hormone usually means endocrine hormones. And that is usually what we're talking about in this class when we say the word hormone. However, these other signaling, exocrine, paracrine, autocrine, the molecules in them are also very hormone-ish and follow many of the same principles as true hormones. So that was the 4 classifications of signaling molecules based on how they are secreted. But what are specifically hormones? And there's a few qualities that make something a good hormone. And those are, it needs to be unique, right? Because it has to fit its receptor and no other receptor. You don't want to get your signals crossed. Like if you're releasing, adrenaline, you want it to have the effects of adrenaline. You don't want some other receptor that looks kind of like it to accidentally respond to it and start doing something else, because you need to be focusing on adrenaline tasks. So they need to be chemically unique, and by preference, they should be cheap and easy to make from something that is already abundantly present. By cheap to make, I mean in terms of energy, because your body uses and makes a lot of hormones. And you don't want to spend a whole bunch of energy making each hormone, or that will eventually sort of start to come out of your energy budget and make your body less efficient. You also want to be able to make them from something that you already have lying around. And you don't want to have too many, because your body has potentially thousands of different types of hormones, neurotransmitters, and signaling molecules. And you don't want to, while each of them needs to be unique, you don't want to make each of them from something different that you have lying around, because you just don't have all that many things abundant in the body. So you want to make them from something that you've already got hanging around. And you want to be able to make them usually pretty quickly. It depends on the type of hormone that you have. Some hormones aren't intended to produce fast responses, but usually you want your hormones to be something that you can make pretty quick. While there are many different classifications of hormones, there are 4 big, broad, common classes. There's a few things that don't fit into these, so you might have an other category as well. These are the 4 most common. First off, we have amine hormones. An amine hormone is something that's made from an amino acid. Amino acids are something that your body has all around, so you can make quite a lot of them. And some of these are made from more than one amino acid that's been kind of cobbled together, 2 or 3 of them that have been hooked in some weird way. A lot of them are the monoamine class. These are amino acids that have had their carboxylic acid removed and then something else put in. So for instance, norepinephrine here is an amino acid that has had its carboxyl group replaced with a benzene ring. This is a benzene ring right here. These are small molecules. They're cheap. They're easy to make. You've got plenty of amino acids hanging around. So they're a very fast, budget-friendly type of hormone. You have peptide hormones. These are short chains of amino acids. Usually less than 20 amino acids hooked together, often with some linker groups in them. All of your cells have ribosomes that make proteins and peptides. They're made from amino acids again. Amino acids are something you have abundantly. And so these, pretty much fit the definition of something that can be produced cheaply, quickly, readily available. A good example of a peptide hormone is oxytocin. Insulin is another peptide hormone. These are things that you could make pretty abundantly, pretty easily. So protein hormones. You might ask, what's the difference between a peptide and a protein? Because they're both basically amino acids linked in a chain. And there are lots of answers to that question, none of them very good. But the main one is that proteins are bigger, and proteins have structure, right? So you can see this hormone here, human growth hormone, has these spirally bits of alpha helices and these sheets. They're beta sheets, and those sheets are folded up against each other into a very specific three-dimensional shape. So once a polypeptide gets big enough that it can assume a specific three-dimensional shape, we usually call it a protein. Protein hormones are not as common because they're not very budget-friendly. Making something that big can take a lot of energy. And the longer a protein, the bigger a protein is, the longer it takes to make. But they do have the advantage that they are very, very, very specific. So human growth hormone is a good example of a protein hormone. Collectively, these three classes are often referred to as the amino acid derivative hormones, right? You're either making something from a single amino acid or a short chain of amino acids or a protein that's made out of lots of amino acids. They're all basically made out of amino acids. Our last category is steroid hormones. Steroid hormones are all derived from cholesterol. Cholesterol is something that is abundantly available. You get it in your food. It's found in every cell in the cell membrane, so it's pretty easy to get hold of cholesterol, and you can modify it in a bunch of different ways. So here we see testosterone over here, and progesterone over here, and there's really just one difference between them. This OH group here is instead replaced with a ketone group over here. And that one small change can make a pretty huge difference. I mean, testosterone and progesterone have very different effects in the body. And these are unique enough that your body can definitely sense the difference between them. Most steroid hormones are end in either sterone or -ol, like estradiol, progesterone, cortisol, that sort of thing. All right, so we have talked about hormones, ligands, receptors, the effects that receptors have. Let's put all of that information together and talk about how receptors and ligands interact and what affects that. So the first key concept that I want to talk to you about is binding affinity. All right? Binding affinity is how well the receptor and the ligand work together. So here you can see this this blue box here. Well, that blue box can move into this receptor, and it will be a nice fit in there. This will have a very high binding affinity. Basically, they match up pretty perfectly. This green sort of pointy-looking thing will fit very well into this green receptor. So that will have a high binding affinity. Now, you could put this square into the green receptor, and it would fit kind of, but it wouldn't match perfectly. And so it would have a lower binding affinity. Binding affinity is a measure of how well two things fit together. Binding affinity is usually measured using a term called KD or KA. So the Ka is what's called the association constant, and Kd is what's called the dissociation constant. And for various reasons, Kd is actually the one that is more commonly used. What you need to know about the affinity constants and or association and dissociation constants is basically if Ka is high, right? So high means good binding. With Kd, since that's a dissociation constant, the higher the number is, the more likely things are to fall apart. That means that the higher the Kd, you're a bad binder. So with Kd, low means good binding. Usually, under a specific set of conditions, like at a specific pH and salt concentration and things like that, the Kd and Ka are constant, right? If you don't change the receptor, you don't change the ligand, you don't change anything about them by altering the environment, then they should remain pretty close to the same over a broad range of concentrations. The next key concept that I have for you is equilibrium, right? We usually think of like chemical reactions as being absolutes, right? A and B combine to form AB, and that's it. When you burn wood, it makes water and carbon dioxide and fire. But in the body, most chemical reactions are actually not just one way. They're reversible. And that means that they are in what's called chemical equilibrium. Now, if you're talking about ligands and receptors, they're in a state of binding equilibrium. They can bind together, or they can fall apart. And in some conditions, you'll find more of them bound together, and in some conditions, you'll have more of them falling apart. So, think of like a dance, right? You've got this big room, right? Like maybe it's a school gymnasium or something, right? And you've got a bunch of people in there. And this is a dance, so many of them are going to be dancing together. So here we have some people kind of dancing together. But at every dance, you also have some people who are kind of by themselves as well. And couples will meet up, they'll stay together for a while, and then they'll separate and go their separate ways, and then you might end up dancing with somebody else. that's sort of the way binding works in this equilibrium context. Here we have A and B, and A and B can bind to form AB, but you see this arrow? You've got a long arrow going to the right and a short arrow going to the left. That means that A and AB, this couple here, can also split apart into just A and B. The way this is structured, right, where you got a long arrow pointing right and a short arrow pointing left, that means that they will spend more time together than they do apart. So that means that they are more likely to bind than to separate. And if you put these three things together, you'll see most of it will be in a B. So maybe this is like 70%. That's just a number that I made up. But, most of it's going to be together. And maybe you have 30% that's separated. Although note that it's not always the same 30%. Some of these A's are going to pair up with B's. Some of these A's are going to pair up with B's, and some of these AB couples are going to break apart. So the dancing metaphor is actually a pretty good one, right? So most people are going to be dancing, but the dances break up, people find new partners, people go get a drink, they go get some food, they go to the bathroom, whatever, then they come back and they find a new partner. Binding interactions between ligands, or hormones, and receptors are all based off of equilibrium. And the more of this AB you have, the more binding you have, the stronger the signal is. The more change it produces in the cell, or the more likely it is to produce a change in the cell. So, since everything is binding according to this equilibrium concept, there are some factors that will actually affect equilibrium. So what are some factors that affect equilibrium? Well, first off, affinity. If you have more affinity between A and B, they bind tighter. Remember that key concept of binding affinity? So if you have a low KD or a high Ka, then they will remain bound longer, and they will spend more time together, and the equilibrium will tilt towards the binding interaction. Going back to our dance metaphor, like if you're with somebody who you're really attracted to, then you're more likely to remain paired up, dance together for longer before you break apart and go do your own thing, right? So, better affinity. So, better affinity leads to more binding. Next, we have concentration, right? So, if you think back to our dance floor metaphor, one thing to keep in mind is that these proteins, they don't have eyes or ears or legs, even really. They float wherever they go randomly. So, in our dance, you can't actually make eye contact with somebody across the stadium and walk right over to them and pair up. You basically just wander around aimlessly until you run into someone. And then when you run into someone, you start dancing with them. So, if you think of our big stadium, if this stadium is like, if it's the size of, a classroom, and you've got like 20 people in it, well, those people are going to meet up with each other pretty quickly. On the other hand, if it's the size of a football field and it's only got 20 people in it, they're going to have to wander aimlessly for a long time before they run into someone. So the more concentrated the ligand is, and the more receptor that there is, the more likely binding is going to happen. So in our equilibrium equation here, A plus B goes to AB. A high concentration would favor the bound state. A low concentration would favor the unbound state. Next, we have temperature. On a molecular level, what temperature means is the speed that molecules are moving at. So higher temperatures mean the molecules are moving faster. So if we think of our stadium analogy, you've got all these people wandering aimlessly around. When they run into someone, they start dancing with them, right? Well, if we raise the temperature, what that means is they're moving a lot faster. If they're moving faster, that means they cover more ground. They're more likely to run into someone. Generally speaking, higher temperatures favor the bound state. Though if the temperature gets too high, it can actually inhibit. But for most temperatures that are compatible with life, higher temperatures favor the bound state. Next, we have contact surface. Contact surface is actually pretty similar to concentration, but one important thing to keep in mind is that most receptors are membrane bound, and so that means that they're not actually wandering around. Your receptors are like on a surface, and you've got your receptors here, and then your ligands are floating free. So the more receptors that you have on the surface, right, then the more likely they are to bind, and that would favor the bound state. That's concentration. But also, since all of your receptors are on the surface, the more surface area that you have, if your cell instead looks like this, and these are your receptors here, well, basically by increasing the surface area, you're increasing the amount of contact that there is between your ligands and your receptors. So greater contact surface area will tend to favor the bound state. Last, there are inhibitors, right? So an inhibitor there are several different types of inhibitors, but one common type of inhibitor is something that's going to basically bind to your receptor, but not activate it and not let anything else bind to it. So here we have our receptor, right? And we've got our ligand. And the ligand wants to get to the receptor, but this receptor is currently bound to an inhibitor, something that isn't activating it, but also is blocking anything else from binding. So let's say, some of the people in our dance metaphor are carrying around drinks. While they're carrying around drinks, they can't dance. They'd spill the drink on themselves. So then the drink is acting as an inhibitor to the dancing. And the first thing they have to do is put down their drink before they can start dancing again. The more drinks you have, the less dancing goes on. So inhibitors will tend to favor the unbound state. Or rather, this one.